hear your stories. But certainly stay connected with us. Anqi did a number of studies, but probably one of the most creative studies she did was in developing methods to characterize bile acids in multiple biospecimens, which really was no small task. And so, as I mentioned, her professor at U of I really did get a gem. I'm really excited to hear about what Anqi's been doing for the last 4 years. It's certainly no short of excellence, because that's the sort of student and professional that she is. So with that, Anqi, let me turn it over to you.
Okay, thank you, Britt, for the kind introduction. And hello, everyone, I'm Anqi. As a master's student who graduated from the program 4 years ago, it's a really honor for me to have this opportunity to share my research to the group. And today I will be talking about the expanding area of health benefits of brassica vegetables. So brassica vegetables such as broccoli, kale, and cabbage, they contain a unique group of compounds called glucosinolates, GSLs. And those compounds themselves, they are not bioactive, but they can be hydrolyzed to form a group of highly bioactive compound called isothiocyanate, ITCs. And this reaction to form the highly bioactive compound requires an enzyme called morosinase. There are two sources for morosinase. One is from plant endogenous morosinase. Another is from the microbes with morosinase-like activities. And later we'll come back to talk about how different sources of morosinase may impact the ITC's activity to the host health. So once the bioactive ITCs are formed, they have been widely studied and well known for their bioactivities in antioxidant activity, anti-inflammation, regulation of metabolic syndrome, anti-cancer, etc. And it's believed to be mainly through the nerve 2 mediated pathway. And these activities and those pathway is right now the major focus in this area of studying how brassica vegetables may benefit the host health. But our group, we are focusing on an expanding area, which is the bitterness of brassica vegetables. And we look at how the bitterness of brassica vegetables may serve as a potential novel underlying mechanism to benefit the host health. So talking about the bitterness, it probably is the least favorable flavor to most people, unless it's in coffee or beer, right? Maybe some of you don't like to eat broccoli or kale a lot because it has this bitter and pungent flavor, especially when they are served in salad, and when you eat it raw, it doesn't have a pleasant flavor. And we believe this bitterness, this unpleasant flavor, comes from the ITCs. So in raw brassica vegetables, the GSLs, they are in the plant stored separately from the plant endogenous morosinase. And when you eat it, the chewing process, it breaks the physical separation between GSL and the plant morosinase. So the GSL get hydrolyzed by the plant morosinase to form ITCs in the mouth when you chew the raw broccoli. And that is when and where you start to taste this unpleasant bitterness in the mouth. However, if you eat it cooked, like if you steam it or stir fry it, the plant morosinase is inactivated by the heating process. So there's no plant morosinase in the mouth when you eat it. So the GSLs won't be hydrolyzed to ITC. So you don't taste bitterness when you eat cooked broccoli or cooked kale. But the bitterness, it can travel to the gut. So we mentioned that there's another source of morosinase that comes from microbes with morosinase-like activities. So when you eat raw broccoli, there are a small amount of GSLs that are not hydrolyzed by plant morosinase. They travel to the lower gut, where they can be hydrolyzed by microbes with morosinase-like activities and release ITCs in the gut. And for the cooked broccoli, the major amount of GSLs, they travel to the lower gut. and the transformation of GSLs to form these bioactive ITCs are mainly dependent on the microbes with marathonase-like activities. So with the microbes that has these marathonase-like activities, they transform GSLs to release these bitter-tasting ITCs in the gut. And once those bitter-tasting compounds show up in the gut, there are some very interesting interaction happening there. So along the GI tract, there are a variety of nutrient sensors spread along the GI tract, including the bitter tasting compound sensors. And for example, if we look at this figure, like those green stars, there could be some bitter tasting compound sensors. They spread along the GI tract at different locations. Some of them located on the enteroendocrine cells, and the enteroendocrine cells, they are endocrine cells in the gut that secrete gut hormones such as GLP-1, CCK, which are very important for regulating the host appetite, food intake, and glucose homeostasis. So we are mainly focused on two bitter tasting sensors in the gut, which is the TRAPA1 channel, which is an ion exchange channel, and the bitter taste receptors. The bitter taste receptors, they are originally identified in mouth, in the taste bud. They are sensors for bitter tasting compounds when you eat and drink something. However, over the past decade, those bitter taste receptors, they are identified in several extraoral organs, such as the GI tract, the liver, and the brain. And that has been attracting increasing research interest in this area. And if we take a closer look at the figure on the right side, for the bitter taste receptor, which is the T2Rs, the T2R agonist, once they activate the bitter taste receptors, it starts a series of signaling cascade, which ultimately leads to increased calcium concentration inside of the enteroendocrine cells. And that will cause the release of the hormone from the cells. And similarly, the activation of TRAPA1 channel will cause an increase in intracellular calcium concentration by bringing the calcium ions outside of the cell to the inside of the cell. And the increased calcium concentration leads to secretion of GLP-1. And the GLP-1 released in the gut can help with regulate the food intake and glucose homeostasis for the host. And that brings to our first research question: does those bitter tasting ITCs released in the gut can interact with the bitter tasting compound sensors in the gut to trigger GLP-1 secretion? So we used an in vitro model to compare the impact of GSL versus ITC on triggering GLP-1 secretion. We also tested a variety of different ITCs impact on GLP-1 secretion. We used this mouse enteroendocrine cell line STC1 cells, and we treated cells with GSL, which is semigreen. It is the major GSL in kale. And for ITCs, we treated cells with AITC, which is semigreen's corresponding ITC. So when semigreen is hydrolyzed by marathonase, it releases AITC. Other ITCs, including BITC and the PITC, they are major ITCs in watercress, and sulforaphane is the major ITC in broccoli. So we treated the cells with those compounds for 2 hours at 37 degrees, and then we collected the cell supernatant to measure the GLP-1 level. And here's the result. We first compared the ITC versus GSL on inducing GLP-1 secretion from the cells. And what we found here is that the GSL, it had no impact to GLP-1 secretion. It's similar to control, which is the clear bar here. But its hydrolyzed product, AITC, it significantly induced GLP-1 secretion from the cells comparing to either control or semigreen, its parent GSL, at all the tested concentrations. So those data suggest that the transformation of GSL to ITC is very important for inducing GLP-1 secretion from the enteroendocrine cells. And then we compared different ITCs, including PITC, BITC, and sulforaphane. For all the ITCs we tested here, and at all tested concentrations, we see significant increase in GLP-1 secretion comparing to the control. So those data suggest that ITC do induce GLP-1 secretion from the enteroendocrine cells. And then we move on to look at the underlying mechanism. Is it through the beta taste receptor signaling pathway or the TRAPA1 channel, or both? So previously we talked about that the activation of either pathway, they ultimately lead to increase of calcium concentration, which cause GLP-1 secretion. So here to study the underlying mechanism, we use several different inhibitors to inhibit the pathway. We use these two inhibitors to inhibit the beta taste receptor signaling pathway, and we use this one to inhibit the TRAPA1 channel involved pathway. And we first look at whether ITC, once they are added to the cells, whether they induce intracellular calcium concentration. So we measure the intracellular calcium concentration using a fluorescent microscope. And the y-axis here is showing the calcium fluorescent signal here. And as we can see, the addition of ITC, the two ITCs we tested here is sulforaphane and AITC. The addition of those ITCs induced a fast and significant peak in intracellular calcium flux comparing to control. We also added the cells with synagrine, which also stimulated a slow and low flux of calcium, but is much lower and slower comparing to the ITCs. So knowing that the AITC do induce the intracellular calcium flux, we added the pathway inhibitors to the cells and see whether the calcium flux will be impaired. So at the figure B is showing that with the addition of the inhibitors, which is the IP3 inhibitor and PLC inhibitor are inhibiting the T2R pathway, and the TRAPA1 inhibitor is inhibiting the TRAPA1 channel. So the addition of those inhibitors significantly reduce the calcium flux caused by AITC. Similarly, we observe some similar reaction here in sulforaphane-treated cells, and that suggests the AITC and sulforaphane, they induce calcium flux inside of the cell through beta taste receptor and TRAPA1 channel involved pathway. After that, we measure the GLP-1 secretion with and without the addition of those inhibitors. And what we found is consistent to the calcium flux result. By adding the inhibitors to the cells, the GLP-1 secretion induced by either AITC or sulforaphane are significantly reduced by those pathway inhibitors. So that is adding the extra evidence to suggest that the ITCs are inducing GLP-1 secretion through those potential two pathways. But to confirm the pathways are really involved in this action, we then knock down the target gene of interest using siRNA assay, siRNA assay. So it is an assay to knock down the gene of interest, and showing here in the first figure, we can see that the siTRAPA1 is the cells that added with siTRAPA1 reagent to knock down the target gene, and the TRAPA1 mRNA expression level is only like 20% comparing to SINT, which is the non-target control. So that suggests there is a success knockdown of the target gene. Once the target gene is successfully knocked down, we treated the cells with AITC and sulforaphane and measured the GLP-1 levels after that. What we're seeing here is that the GLP-1 levels are significantly reduced when the TRAPA1 gene are knocked down in those cells, in both AITC treated cells and also the sulforaphane treated cells. So that suggests the TRAPA1 channel is involved in both AITC and sulforaphane-induced GLP-1 secretion action here. And then we knock down another target gene, which is the beta-taste receptor member 138. It's important to know that there are 25 beta-taste receptor members identified in human, and there are 35 different beta-taste receptor members identified in mouse. And we only chose this specific beta-taste receptor knockdown is because first, this receptor is the most studied one. And also AITC has been found to be able to activate this beta-taste receptor member. What is unknown is whether the GLP-1 induced by AITC is via this receptor involved pathway. So we knocked down this target gene and we treated the cells with AITC or sulforaphane. What we observed here is that the GLP-1 secretion induced by AITC is reduced by knockdown the beta-taste receptor 138. But the GLP-1 secretion induced by sulforaphane is not impacted by knocking down this beta-taste receptor member. So this is interesting because it is suggesting that sulforaphane is inducing GLP-1 secretion through maybe other member beta-taste receptor members, but not this one, not the 138 that we're studying here. So in conclusion, for the in vitro study, the transformation for GSL to ITC is very important for inducing GLP-1 secretion. And AITC-induced GLP-1 secretion via TRAPA1 and beta-taste receptor 138 involved pathways, but sulforaphane-induced GLP-1 secretion via TRAPA1 involved pathway, but not beta-taste receptor 138. So why is those findings significant? It is suggesting that the ITCs released in the gut may interact with the beta-tasting receptors in the gut to induce GLP-1 secretion from the gut, and that could be beneficial to the host metabolic health. So you may wonder if we can confirm that in vivo, and I will talk about that in a later slide. But right now, let's take a step back and look at how dietary GSLs may modify the microbial metabolism of this compound. We talked about that the GSLs, once they travel to the lower gut, they can be hydrolyzed by microbial marathonase to form bioactive ITCs. However, the reality is much more complicated than this. The microbes, they can not only form bioactive ITCs, they can also transform this compound to some inactive compounds, such as nitrile or disulfo GSL. And the pure ITCs, once they are formed, they can also be conjugated in the host to form the glutathione conjugate or ITC cysteine conjugate. So there is a much more variety of microbial metabolites of GSL than just only ITCs. And we're interested at looking at how broccoli consumption may alter the gut microbes to change the microbial metabolism of GSL and see if it's possible to shift the direction to form more bioactive ITC instead of other compounds. So to answer that question, we designed this short-term cooked broccoli feeding study. We fed both lean and obese mice with cooked broccoli or control diet for continuously 7 days. And at the end of the intervention, we collected their cecal content and used that to study the microbial metabolism of GSL. What we did is that we collected the cecum from the mice, and we immediately transfer the cecum into the anaerobic chamber, and we collect the cecal content, incubate it with GRP, which is the major GSL in broccoli, anaerobically for 2 hours. After the incubation, we collect the supernatant and use LCMS to measure the metabolite profile and to study how the GSL metabolism are changed. by broccoli consumption. And here is the result. We can see that in both lean and obese mice, if we first look at sulforaphane, which is the free ITC hydrolyzed from GSL, in both lean and obese group, the mice fed with broccoli supplemented diet, the cecum content are produce much more higher free ITC when they are incubated with GRP in the anaerobic chamber, comparing to the control. In the control group, the ITC level is lower than the detection limit. And similar for the sulforaphane cysteine conjugate, we observed a significantly higher level of these metabolites in the cecal content incubated with GRP when they are from the mouse that are fed with cooked broccoli. Same for total ITC. So for the measurement of total ITC is another method. It's a reaction called cyclocondensation. It transforms all the ITC in all form, not only free, but also the conjugated ITC to one compound. And we measure that compound, consider it as the total ITC. So the total ITC is also significantly increased in the cooked broccoli fed animal comparing to the control. We also observed an increase in nitrile production by the cecal content from the mice that fed with cooked broccoli. There is a trend of increase, but not statistically different. So those data suggest frequent cooked broccoli consumption can result in increased ITC production by the cecal microbiota. We also measured the ITC's bioaction in the gut, which is the NQO1 activity. We found that the increased ITC production in the gut also led to the increased antioxidant activity in the gut, suggesting that the increased ITC production also had increased bioactivity there. We also looked at the microbes that are associated with those microbial metabolites. And we found several interesting groups that are significantly associated with those metabolites. And right now we're still at an early stage of analyzing this data. And I'm showing the microbes at the genus level, but hopefully we will be able to find the microbes at species level in the upcoming couple of months. And hopefully we'll be able to find some isolates and use those to further study how those microbes metabolize GSL to form either bioactive or non-bioactive metabolites. So the conclusion for this short-term cooked broccoli study is that frequent cooked broccoli consumption, it alters mouse cecal microbiota to improve GSL hydrolysis to bioactive ITC in both lean and obese population. And now it comes to the question, does the increased intestinal ITC production impact the GLP-1 secretion and thus the host metabolic health? Our hypothesis is that eating raw versus cooked broccoli, it leads to the release of ITC at different locations along the GI tract. And once the releasing location is different, they may interact with the local bitter tasting compound sensors, and that cause the release of the secretion of GLP-1 at different location of the GI tract. And we wonder whether that will lead to different metabolic health impact to the host. And to answer that questions, we designed this 4-week study. We used diet-induced obese mice. So those mice are already fed before we start feeding them broccoli. And we fed them with high-fat diet with cooked broccoli, or raw broccoli, or with concentrated GRP powder. So the concentrated GRP powder, the level of GRP are matched with the cooked broccoli. We fed them for 4 weeks, and the food intake, body weight are monitored weekly. we measured their fasting blood glucose, insulin, GLP-1 levels at the end, and lipid profile at the end of the study. We conducted oral glucose tolerance test at the end of the study. We also collected the GI tissue for gene expression, liver and pancreas tissue, fat tissue for gene expression. We collected sickle content for microbiome analysis. So looking at the food intake, which is monitored weekly, we did not see overall treatment effect on food intake. But what we can see here, there is reduce of food intake at the first week in the raw broccoli group. And we think that is because the animals are adapting to the new diet. And obviously they don't like raw broccoli either. It tastes bitter, but they are slowly getting adapted to the diet. And starting from the week 2, there's no difference in food intake. And as for body weight change, we observed a reduced body weight gain in the raw broccoli group comparing to other groups, but there's no difference between other groups comparing to control. And for oral glucose tolerance test, we conducted at the end of the study, and we did not see any changes in oral glucose tolerance test or the area under the curve of OGTT. We also looked at the fasting glucose, fasting insulin level, C-peptide level, and also calculated the HOMA-IR score. What we're seeing here is that there's no difference in fasting glucose level among the groups. And for the fasting insulin, there is a trend of reduced insulin level in the treatment group comparing to the control group, especially for the raw broccoli group, but there is no statistical difference here. Similar for C-peptide. C-peptide is a protein that's secreted together with insulin because it's more stable than insulin, so people use it as a marker for insulin secretion. And we measured this peptide is lower than in raw broccoli group comparing to other groups, but again, there's no statistical significance here. Similar for HOMA-IR score. We measured fasting lipid profile, and this time we do see a significant change here for the raw broccoli group. It showed a reduced LDL level in the raw broccoli group. And for the triglycerides, there are trend of reduction, but not significant. And then comes to the postprandial GLP-1. We did not see changes in postprandial GLP-1 either among the groups. And one reason is that we found it challenging to measure postprandial GLP-1 in mice. The way we did it is that we fasted the mice during the daytime for 6 hours, and then we provide the mouse to free access to food for 30 minutes. And after 30 minutes, we remove the food and we collect the blood from the tail vein. But while we do the experiment, we found that it is hard to control the mice, to let them to eat at the same time and let them eat the same amount of food during the time we want them to eat. So we found that some mouse are eating right after we give them the food, some are eating only at the last minute during the 30-minute time period. Some are eating more, some are eating less. So it's hard to control the time and the amount of food that they eat during this period. That's why this postprandial GLP-1 result is kind of hard to interpret here. But based on the result, we did not see any changes in GLP-1 secretion. We measured the gene expression along the GI tract at upper versus lower GI tract. The duodenum is the upper small intestine, and colon, we used the distal colon tissue to measure the gene expression. For the GCG gene is the gene responsible for GLP-1 secretion, and we see a trend of increased GCG gene expression in the RB group, which is the raw broccoli group. But when it goes to the colon, we see a trend of increased GLP-1 secretion gene in the CB group and GRP group. So that is kind of makes sense because our hypothesis is that in the raw broccoli group, the ITCs are released at the upper GI tract to induce GLP-1 secretion, whereas for the cooked broccoli and the GRP compound, the GSL group, they goes to the lower gut where the ITCs are hydrolyzed by the microbes to release in the lower gut to trigger GLP-1 secretion. And similarly for the beta taste receptor genes that we measured here, we see a trend of increased gene expression for those beta taste receptor members at the upper GI tract in the raw broccoli group, whereas at the lower GI tract, we see a trend of increased gene expression in the cooked broccoli group and the GRP supplemented group. For the TRAP-A1, we did not see any significant change in the expression, but one reason is that the gene expression in the GI tract is relatively low, and the data quality is not quite well. And as you can see here, we only have 3 groups of data here. So, but overall, those gene expression data suggesting that our hypothesis that the raw broccoli consumption release ITC at the upper GI tract to activate beta tasting compound sensors, cause GLP-1 release, and the cooked broccoli group, they release ITC at lower gut to interact with the beta tasting compound sensors at that location. So in conclusion, the frequent broccoli consumption improve the efficiency of gut microbial transformation of GSL to bioactive ITC in both lean and obese mice. And the 4-week raw broccoli consumption reduced body weight, fasting LDL in diet-induced obese mice, also showed a trend of reducing fasting insulin, C-peptide, triglyceride, and the HOMA-IR score. And eating raw versus cooked broccoli resulted in releasing of ITC at upper versus lower gut, led to trend of increased GLP-1, T2R, and TRAP-A1 gene expression at the ITC releasing location. So overall, we found that eating broccoli, it results in the gene expression difference along the GI tract depends on the type of broccoli you eat. And hopefully in the future studies, we will be able to detect the changes in GLP-1 secretion. And that is the end of my presentation. Thank you everyone for your time and attention. And I'm happy to take any questions.
All right, thank you. That was great, Anqi. Do we have any questions from our audience?
I do.
All right, John, you're on.
So first of all, I'd like to congratulate you on developing new assays. I think that for any good discovery, you really need to learn how to measure compounds accurately, and it's through that ability to measure compounds that you actually make progress in science. So I think that's a fantastic contribution you've already made in your short career. Regarding taste receptors, though, rodents are really interesting. Rodents will modify their taste receptors and their salivary proteins in response to diet. And clearly you've changed your diet, at least in the mouth. Is that also true in the intestinal tract? Do we understand what those changes can be? Because at least in the mouth, it's well documented that the salivary proteins which bind with these bitter compounds, by the way, and the taste receptors will modulate with change in diet. So how might that affect if it also changes in the gut?
Well, that's a good question. I think currently it's unknown whether the diet will change or modify the expression of those genes in the gut. And for example, in human, some people are like for the bitter tasting. Some people are tasters, some people are non-tasters, and some are super tasters. And we don't know whether their taste sensitivity in the mouth are also different when it goes to the gut. And that is definitely a very interesting area to look into in the future. But yeah, sorry, the current answer is I don't know.
Okay, thanks.
Well, Anqi, that was great, because one of my questions was going to be about these super tasters, so I'm not going to ask that. Do we have any other questions in the group? We have
Yeah,
plenty.
I have one quick question. I probably missed this part, but what's the dose used in the in vitro study, and how would that convert to human consumption if we want to achieve the same result?
Yeah. So the dose that were used in in vitro study, they are physically relevant. They are actually lower dose than the dose of ITCs that occur in the GI tract after consumption of broccoli.
Mm-hmm. So like what?
After a consumption of 1 serving of broccoli in the GI tract, the level of those ITCs in the GI tissue is higher than the concentration we tested here.
Okay. Thank you.
Thank you.
Others? Well, while people are thinking, Anqi, I'm going to ask you a question relative to your GLP-1 results. So, these gut peptides I, absolutely love. And you may or may not know that CCK was what I spent quite a bit of time doing my PhD on. And then GLP-1 came around years later, and we started adding that to our battery of analyses. So this is really fun stuff. So one of the questions I have for you is, knowing that these gut peptides can be very fussy when you analyze them, and they also have, active molecules and inactive molecules. And so my first question is, when you collected from your mice, did you protect the sample?
Yeah, we add the DPP-4 inhibitor to protect the sample. And we measured total GLP-1 for the sample we collected from mice. But for the cell culture study, we only measured the active GLP-1.
Ah, okay. And so do you think, especially since you got the gene expression results that didn't predict what you might have seen in the plasma, that maybe that's the difference between the total and the active assay that you chose? Maybe?
Yeah, it's hard to measure only active GLP-1 in mice, especially when the GLP-1 after its secretion is very unstable. And so, yeah, I guess.
And you only have so much blood too, right?
Yeah, we only use 5 microliter for the analysis for each sample.
Wow.
Yeah. Well, and then clearly there's something going on there. I mean, and that's another reason to think about the active part of this. Not that GLP-1 is the only molecule to be thinking about. It's a really good one. But your energy, your food intake, and your body weight data suggests that there's an energy expenditure effect, right? Because your food intake came back up. Were those animals, did you notice by chance if they were more active in the in their cages?
We didn't measure the energy expenditure, but just by observation, I don't think there are a difference. And they are single caged.
Yeah. Okay. So yeah. So maybe maybe this is, another, clearly something's happening. These guys aren't gaining as much weight, and it's really intriguing. So thanks. Others?
Thank you.
Come on, guys. Who's out there? I gotta look at all these faces. All right. Nupur, do you have any questions for our speaker? No? Okay. Okay, so I do have a question then for you, Anqi. Nitrile. So I think in one of your, I don't know what slide it was on, but your nitrile, you had right there. You had some increases in your control. So what is the source of that, do you think? Am I reading that correctly?
It's like nitrile level is like higher than the other ITC compound, so it's it's higher than the detection limit. That's why we were able to measure it. But for the ITCs, they are lower than the detection limit, so they are showing like nothing here. But
I see.
yeah, it's not like a increase in the control.
It's just
the,
that, okay.
the detective issue.
Gotcha. Okay. Very good. Well, this is your practice for your defense, right?
Yeah.
No. All right. Well, we could say thank you to Anqi, but I'll make one last round of, does anybody have a burning question? Okay, Anqi, I guess you're off the hook. It's really, I'm so glad you joined us today. Really good to see you. Really excited about your future, and many congratulations from your family at Illinois Tech.
Thank you. Yeah, it's really nice to see everyone here.
Yeah.
And Among, hi Among, I didn't get to talk to you. How are you?
I'm good. How are you?
I'm good.
I thought you would be busy in presenting. We can chat later. Nice
Maybe
presentation.
when we're not recording all the conversations.
Yep. Nice presentation, and good luck with your new venture. I'm pretty sure you'll do great wherever you go.
Thank you. Thank you.
All right. Well, that takes care of our Thursday seminar. Thanks, Anqi. Stay in touch, okay?